Hello and welcome to RMBL's Remotely Curious About Science series. Tonight we are talking with Dr. Rosemary Smith from Idaho State University, who studies some really cool beetles. So I'm going to turn it over to her, and then we'll have times for questions that you can put into the chat function, and we'll watch that and give those to Rosemary as we go. So I'll take it away, Rosemary.
Great. Well, thank you and welcome to all of you for joining us today for the RMBL Field Science Challenge #2, the mystery critters in our midst. And I'm Rosemary Smith. I'm a recently retired professor in biological sciences at Idaho State University. And I have a long-term association with the Rocky Mountain Biological Laboratory. I first went out there as a college student, and then I did my master's and PhD research out there in the field of behavioral ecology. And if you did last week's Field Science Challenge and you learned about golden mantle ground squirrels, that was actually the subject of my PhD dissertation. So I have a fondness for those creatures as well. And then since the early 90s, I've been studying the burying beetle, which is the subject of today's challenge. So I hope that you were able to watch the little 4 or 5 minute video that showed my students doing field work, trapping the burying beetles. You could see them live and also look at some of their interesting features of their antennae and their body coloration. I'll have have a few pictures for to show you about the burying beetle, but I thought I'd start a little bit with who they are, where they get their name, and what kinds of things, why they're sort of interesting, why people have been really intrigued by their behaviors. So let's take a look. Oops. Erin, can you move my screen? Oh, there we go. So the key feature of the behavior of the burying beetle, where they get their name, is that they bury small dead animals underground, and they use that buried carcass as a resource to feed their developing larvae. So they have fairly extensive parental care where they both provision their offspring, but also feed them throughout development and stay with them until they've completely finished feeding on the carcass. So if you look at that kind of cartoon on the right of the screen, it shows the mouse being buried by a pair of beetles, being rolled up into a ball. And then while they're in a little chamber underground, the parents feed the young. One of the intriguing pieces of the burial behavior is also that they have what's called biparental care. So that both parents, both the male and the female, the mom and the dad, care for the young. And that's fairly unusual in the animal kingdom of having biparental care. There are examples, especially you're probably most familiar with birds, where both the male and the female might feed the young at the nest. And it seems, in fact, to have evolved primarily in organisms where the provisioning can be done equally by both parents. So in insects or in birds, provisioning might occur by both males and females. In mammals, in contrast, the provisioning or the nursing of the young is a female characteristic. And so males often can't assist with rearing the young because of not having the ability to feed the young directly. They could provision the females though, which is what they do, for example, in wolves or a few of the social carnivores. But for the most part, most mammals, there's just maternal care. So biparental care in itself is interesting, and when and where it evolves. So I've been interested in studying the behavior of the burying beetles for a number of years, and I've done all kinds of different studies, both with my students. I've had probably over 40 or 50 undergraduates who have done research projects on the burying beetle, and we've looked at everything from burial and carcass preparation. We've looked a lot at the parental care of the larvae. We've looked at flight and thermoregulation, which is regulating their body temperatures, communication. So Omar, who's on the Zoom call right now, he studied communication in the beetle. They make a kind of a squeaking sound, a stridulation. And we've studied a little bit about when and where they use the stridulation call, and also a lot with different interactions between species, between flies and ants and other species of beetle. So it's kind of a cool system because there's so many different behaviors that are intriguing and that are worthwhile to study that we really don't know that much about them. So this challenge, the challenge that we have for you this week, is following up, not so much looking at the behaviors of the burial of the carcass, so you don't have to actually have a carcass handy, but rather to really study more about their ecology. And broadly, ecology is the study of the distribution and abundance of species. So where do they live, and where they live, how common are they? Are they fairly abundant, or are they fairly rare? So if we can answer those questions about a species, then we can understand a lot about its ecology. So the burying beetles are in the genus Nicrophorus. Nicro is dead and phorus is moving. So burying or moving the dead is where they get their name. And if you look at this weird map on the right, you'll see that it's actually looking down on the North Pole. And if you look down from the North Pole at map of the Earth, what you can see is that most of the dots are around the pole. So they have a northern distribution. They're not found south of the equator. So they're in northern temperate areas, Eurasia. There's only 2 common species in Colorado, in the study area where we work at in the Rocky Mountain Biological Lab. That's in Nicrophorus investigator and Nicrophorus diffodiens. But there's 2 other species that we do occasionally capture that are rare and that are new occurrences in the study area. So one of the things that we're really interested in engaging citizen scientists like yourselves is to try to find out where these 2 other species are living, where they are common or abundant, and whether that is a particular habitat that they're in or a particular elevation that they're at. So we know that Nicrophorus cutula, for example, is more desiccation resistant than Nicrophorus diffodiens or Nicrophorus investigator. So it's possible that they are more abundant in the Almont Gunnison area, in the sagebrush, which is hotter and drier, and that they are only increasing in the kind of warmer, drier areas, moving higher up in elevation. But we don't know that. So we would love it if you guys would help us in this challenge to capture some beetles and let us know where these 4 species are. Are they in your backyard or not? That's kind of what we want to find out. Are they around where you're living? We know who's at Rumble, but we don't know of the rest of either the East River Valley or Colorado or anywhere you are, as long as you're in North Temperate North America or Northern Europe and watching this video. Anywhere north of the equator, you have some chance of catching the Nicrophorus. It would be really interesting to get more data on their distribution and abundance. So how are you going to go about that? How can you actually catch a beetle and figure out its species? So I've got a couple slides so that we can work on this challenge together. The easiest way is to catch them by using a bait. And the bait is their favorite food. Their favorite food is meat. So any kind of meat, they're very carnivorous. We use chicken legs because they are easy to buy at the store. They're relatively inexpensive compared to other cuts of meat. And they don't like instantly rot like ground beef does. So they actually can stay in a baited can for a week and not be too bad in terms of rotting. Omar might disagree there, but not too bad. And so you would place, to build a trap to catch one of these beetles, you just need a can. You can also use a 5-gallon bucket like the kind you get at Home Depot. You can use a gallon ice cream container, plastic bucket. You can use a smaller container, but it might get too hot. So you want to have it a big enough volume that it just doesn't turn into like a little oven. So you get your can, poke some holes in the bottom, put a little soil or potting soil or peat moss or leaves, anything in the bottom to kind of keep some moisture in the bottom. And then put your bait in and then cover it with a screen top. There's pretty good descriptions of what the whole can is going to look like in the written description to this challenge. So if you're having problems building your own trap, you can refer to that guide in the challenge, and then you'd want to put it outside. We normally put ours on a stake, and that's just to keep the smaller scavenging animals away from the chicken. So for example, it might keep away a weasel or another sort of a small scavenger. It won't keep out like a bear or a large dog or a fox. So if you are in a location where you're likely to get a large scavenger, then I would strongly suggest that you either put it on a taller fence post or on a tree, like at least shoulder height. and that will keep it away from most of your foxes and coyotes and dogs. Pretty much nothing will keep it away from a bear or a pine marten. So you might just lose your chicken leg to a very large carnivore, but most likely you won't have anybody coming to the traps except for burying beetles and some flies. So usually the question is, how long do I leave it out? We usually put the chicken out, and usually within a couple days, certainly 3 or 4 days, you will have, if the beetles are active, you will have lots of beetles. Another question that I often get is, well, how many beetles am I going to get in my trap? And really you can get no beetles, or you could get, even in one trap, you could get up to 40 beetles. So it really depends, maybe even more. It depends how active they are, if it's been really good flying weather for them, so sort of warmer temperatures, and if they're active at that season, if you put it out in a site that they're abundant, then they will definitely aggregate in your trap. So what do we want you to do with them? If you peer into your trap and you see one of these orange and black beetles, they're large. I put this picture in here with the hand so that you can see they're a fairly large beetle. You can just pick them up, and then what we'd like you to do is count them. That would be helpful. And then if you want to give your hand at identifying them, I've got a little couple pictures for you to look at to see how to identify them. The easiest way for the ones in Colorado, at least at the around RMBL, but anywhere will work, and we'll send you a key for some of the other species, is to look at their antennae. Their antennae are one of their most interesting characteristics. You can see they're large. They have big balls at the end of their antennae, and in some species, the balls at the end of their antennae are solid black, and that's in Nicrophorus diphodians. So if you look at the ball and on the end of the antennae and it's solid black, it's most definitely, if you're in Western North America, it's Nicrophorus diphodians. If it's an orange ball at the end, then it could be any number of species. The two most common ones are Nicrophorus gutula and Nicrophorus investigator. And the way you would tell those apart is by looking at the edge or the side of the hard outer wing cover. And I put some white arrows there on the screen. If you look at the edge, and you see that there's a lot of black on the edge, then it's probably Nicrophorus gutula. If you look on the edge and it's solid orange, then it's probably Nicrophorus investigator. If you can take a picture of your beetle that you capture from the back side and also from the side, like with the pictures that I have there, then I can help you identify them if you post them to iNaturalist, which you learned about in last week's challenge. So, and then we will also have a link to a key, an online picture key, kind of like the pictures that I'm showing you here, where you can go through, but with even more species. So that key will be posted on the challenge after this talk. So that'll help you. Then if you catch, so now you've made a trap, you've left it out for a few days with some good bait, and then you've peered in, you've seen some beetles, you've pulled them out, and really what we would love you to be able to do is tell what species. If you can't, just count up, like I caught 6 or I caught 10. That would be really great. If you can tell, like I caught 6 with orange antennae and 2 with black antennae, that would be even better. And then if you can separate the ones with the orange antennae into different species, that'd be great. So any level that you feel of identification is fine. So just kind of going to pause there and just sort of end up saying, people often ask me, like, well, where should I put my trap? So one of the things that we're interested in is how they use the different environments. You don't have to go all over the world like I showed you the world map, although we'll be able to collect data from different people in different locations. Maybe we'll be able to see different elevations. But even within an elevation, there are different, what we call microsites. So there's different habitats within a site. So if you look at these photos, you can see there's an open meadow, but there's also aspen forest, and there's also spruce forest, and there's also rock cliff face. So we could also try to find out whether or not the beetles are, what species are in some of those different microhabitats that exist within a fairly small region. So you could also give your trap location as being in the aspen forest, or your trap location being in a spruce forest or an open meadow. And that would be interesting, useful information as well. So I think I'm gonna pause there and see if there are any questions. That just gave you a brief overview of what this challenge is. And then I also have, later I can show you some of the data that we've collected over the years, but I'm happy to answer any questions that you have right now. Are there anything, Erin?
I don't see any right now, but I was wondering, when did you first see Gutula? I worked with you at one point and I never saw one of those before.
So that's great. I actually have that on a graph. See if I can. So here's a graph, and Gutula is in the red line at the bottom of the graph. And you can see from 1995 to 2005, we never caught a Gutula. So I don't remember what years, but you were in there somewhere. And then after 2000 and about 6 or 7, we caught very few. And ever since then, we've caught them every summer, but never in very high abundance. But we have caught them every single summer since then. So they did, and then one of my students did his master's thesis on where Gutula are more common. And if you look at the map on the right, to orient some of you who are in the Crested Butte area, right in the middle of the map is Crested Butte. The mountains are in that kind of blue color, and the yellow are the valleys. Gothic is at the end of that blue arrow, and down at the very bottom of the map is, you can see the word Almont. And if you just look at the red, that's for Nycrophoris gutula. The size of the pie slice is the proportion of the beetles that were gutulas. So you can see that the red pie slice is much bigger, closer to Almont. And then it kind of dwindles as you go to CV South. There's a few in CV South. And when you get up to Gothic, they're very, very rare. And when you go up past Gothic, we've never caught any. Like we've never caught any up in Bellevue or Upper Washington Gulch. So that's one of the things that if you're doing this challenge and you live in one of the areas like near Almont or CV South or CB or Washington Gulch, that would be really cool because we haven't done that survey since 2015. So it'd be really nice to update that. Good question.
Have you seen any variation in the numbers of those that you catch with different snowpack? You said they have a higher resistance to desiccation. Have you seen them more in drier years or no?
We haven't done that analysis. That would be a great analysis to run, is to see whether or not they're more common in, based on whether it was like a longer, hotter summer or gave them more chance to fly to Gothic, or whether they just were able to survive a winter better if it was shorter. So yeah, we've not done that analysis, but that would be really interesting to kind of put together kind of what physiological factors or temperature, moisture factors are important for their abundance that would influence their abundance.
And Daniel asked if it seems like Gutula are moving to higher altitudes over time.
So that's why this study would be great to repeat, because as I said, it was done in like 2014, 15, and we have not followed up at all of the other sites that Kevin did his master's thesis at. So we haven't gone, so every place where there's a circle there, a pie, we haven't gone back to those sites. So it would be really interesting to see if, let's say, then a number of Gutula at the sites where it was either absent or very low, whether it's increased. That would make a really great study.
Mm-hmm. And Anne is wondering how you became interested in studying the beetles, and what is something that has surprised you about over your years of research on them?
So you can tell from this graph that we have data from 1996. So that is one of, I actually started studying them a few years before that, but I didn't have enough data to really make much out of it. But I became interested in them based on teaching a course at RMBL on animal behavior. And we were interested just in the basic biology of parental care and how organisms choose places to rear their young. And so we were looking at where nests were located. And because that's a choice, so where do butterflies lay their nests or egg laying sites, where do birds lay their eggs, where do butterflies lay their eggs? And then I had just heard of the burying beetles, and so we threw out mouse carcasses, which are actually very common because there's usually mice in the cabins at Rumble, so we were all able to get mouse carcasses, and we put them out in different habitats at RMBL, and just asked the question of where the beetles are located, if they even existed at RMBL, because we didn't have any collected before there. And then they showed up, and they were so interesting that my students and I, the whole summer, that's all we did for the whole rest of the summer, was do burying beetle experiments. And it kind of just took off from there. They're really interesting organisms if you get to see them alive. So.
And what's been the most surprising thing that you've learned about burying beetles? A lot to choose from.
Yeah, there's like, I liked Omar's study. Last summer we were studying the stridulation. They have kind of a rasp and a file on their wings where they rub against their abdomen. It makes a squeaky sound, and we knew that they used the squeaking sound for communication with their young. So they use that call to when they have food available. So in this picture here, you can see there's an adult up on a carcass that's been rolled into a ball, and there's some tiny larvae inside the ball. When they're ready to feed them, they make a squeaking sound, and the larvae kind of rear up and open their mouths, and then the parents feed them directly into their mouths. And so we knew they used the squeaking call for that because you can hear them using it while they're feeding the larvae. So that one was pretty obvious, but we thought that they probably used the squeaking call between adults. And so Omar, in his study last summer, was able to actually document with a video and sound recording of them using it when they were moving the carcass, which was really neat because that sort of suggests a certain level of using that sound to collaborate on a shared task of moving the carcass, because they could drag the carcass like a meter to some soft soil or wherever they want to bury it. They don't have to bury it wherever the carcass is. They can kind of move it around, and when they move it, they seem to squeak to each other. So that was kind of cool to find that out. That was kind of a neat discovery.
And Omar says he has a link to a video of that.
Oh, great, great.
Put it on the website too.
Yeah, well, definitely, that would be cool. I mean, they're always interesting. One thing that we thought is that there's no way the larvae could survive without their parents. So another undergraduate several years ago did the experiment of what happens if the parents are removed. And we thought that they needed the two parents to survive, but it turned out that they really only needed one parent was fine. And that single moms were actually better than single dads, but single dads were fine. They were perfectly capable of rearing the young on their own. The surprising thing in that experiment was that some of the larvae were able to survive without either parent, but they had to be a certain age. They couldn't be like really, really tiny. They had to have been already a couple days old. So it seemed like the parental care was most important when they're tiny. And then as they get a little bit bigger, if their parents disappear, they're still able to consume the carcass on their own.
And you think that's just mouth parts related?
Yeah, probably when they first hatch, their mouth parts aren't hard enough to dig through the meat, because meat is kind of tough. They might also have to get you're probably familiar how we all have a gut microbiome. The beetles also have a gut microbiome, so they might also have to get a gut microbiome from their parents, because their parents do feed them directly from their own mouth. And so they might have to get those bacteria, those good bacteria, into their stomachs for them to develop properly. So it's also possible that without feeding from the adult that they won't get the right microbiome. But we haven't done that experiment, but that's a hypothesis.
And Venice is wondering, do you bury the mice for them, or do you let them do all the work?
Oh no, we let them do all the work. It's really fascinating to watch them do it. I mean, they can easily do it in just a matter of hours. We've done a number of experiments seeing like how fast they can do it. whether the soil itself makes any difference. Like if it's really loose, light soil, they can obviously bury more easily than if it's really compact, or if it's rocky. A lot of times if it's in a bad environment, like outside, if it's like on a really rocky, gravelly area, they'll just drag the carcass to some softer soil at the base of a plant. So we've done experiments both outside where we let them bury it just like naturally, and then they tell us where they want to bury the carcass. We've also given them choices in the lab in a container. And then we can also watch them. We use clear plastic containers to have them breed in the lab, and then you can see the burial, and then you can kind of uncover it and see the larvae in that little kind of cavern. So like these pictures here would be one that we had uncovered out of a box, probably. Yeah, I don't know. I think I have a, yeah, so here in this picture, you can see the larvae inside the carcass. There's kind of the tail is going off to the right. And the two adults are on the carcass, and the larvae are inside. So when we do it in the lab, we can kind of slightly uncover the soil, and we can see the larvae and the adults on the carcass. So Vanessa, we'll do that experiment this summer.
And how do they overwinter?
Oh, so they overwinter as non-feeding larvae. So the life cycle of beetles are similar to butterflies. They have complete metamorphosis. So they have the different life stages that don't look alike. So they have the larva, like in butterflies, it would be a caterpillar. In beetles, it's called a grub. And then that's the feedings, usually the major feeding and growth stage of the organism. They grow as big as they can. In the beetles, they consume the entire carcass, and then they rest for a while, and then they pupate. So they turn into the pupa, which is also a resting form. It does not feed. And during the pupal stage, they metamorphose or change into the adult. And the adult is the stage of the insect that has the wings and reproductive parts. So in butterflies, that would be the adult stage. And in the beetle that you see as a beetle is the adult stage, and it's winged. And so that cycle can either, the Nycrophorus investigators, so the species with the orange antennae, they overwinter as prepupa or non-feeding larva, and then they pupate in June right now, and they emerge as adults in late June. Nycrophorus diphodians, the one with the black antennae, they actually pupate in late August and September. They emerge as adults, and then they find a place to overwinter as an adult. And then they're some of the earliest that emerge in the spring because they've already metamorphosed. So they don't have to go through that pupation and metamorphosis because they did that in the fall. So they come out earliest. So usually when we catch our most diphodians is in late June and early July. So I guess the answer is they have different strategies. Some might overwinter as adults, and some would overwinter as larvae.
But even the adults overwinter underground, like, do they still?
Yeah, they would all, so the larvae would overwinter in the soil, probably at least 10 to 15 centimeters below the surface of the soil. We've done a number of overwinter experiments where we place them in containers in the ground over the winter, and the RMBL area is sufficient because the snow cover is deep enough that it keeps the ground from freezing. So we usually bury recording thermometers with the beetles, and where it's always about 4 degrees C, which 0 degrees C is freezing. So it's just a little bit above freezing pretty much the whole winter at about 15 to 20 centimeters below ground. So they don't freeze in the winter.
Cool. Do we have any other questions? Did you have other things you wanted to talk about, Rosemary? Or that you had slides for? I didn't know if we made it.
Yeah, I have, I mean, a couple other, I was going to highlight a couple of the other experiments that I have, unless there's more questions. Okay. So one of the studies that we've been doing that, again, is the ecology of, Erin, did you say something? No, is the ecology of the species. Is trying to figure out the numbers. So remember, I said it was the distribution, where are the beetles, but also the abundance of beetles. So trying to understand why are the beetles, some years there's a lot, and some years there's very few, and trying to understand that. We know that the beetle reproduction is entirely dependent on mouse carcasses. So it makes sense that the number of beetles would be related to the number of mice. So since 1996, on the same sites, we have been capturing beetles as well as mice to get the numbers, the abundance of the mouse carcasses that might be available, as well as the beetle carcasses. So this is just a graph of one of our sites of each year that we've gone out and calculated about the number of beetles. So you see, like, on the x-axis is the year from 1996 to last year, and then on the y-axis is the number of beetles. The first thing that, or the number of rodents. The first thing that you can see is that there's about, where it's like the beetles are in the thousands and the rodents are in the hundreds. Okay, beetles are much smaller. You can get a brood of like 20 beetles out of one dead mouse. So it's not surprising there would be, in the food chain, there would be more beetles than mice. So that's the first thing. Then if you look at that pattern, it just kind of looks random, like it's just going up and down. Like clearly some years there's a lot of rodents and some years there's very few. Some years there's a lot of beetles and some years there's not very few. The other thing you probably notice is that the little peaks, they don't really line up. Like if there's a peak of rodent, there's not a peak of beetle. So that doesn't really make sense, except that that's not really the right way to think about it. So the right way to think about it is that the number of adult beetles that we capture actually were born last year on last year's rodents. So we need to offset our curves by 1 year. So see if there's more rodents 1 year, if there's more beetles the next year, because they would be coming out having bred or been born on the rodents last year. So that's a different way of graphing it. And that's this graph here. So we have the rodents, it's not numbers now, but it's biomass. So it's how many grams of rodents were out there. And then the so that's the rodent biomass on the x-axis. And then on the y-axis is the population size of the Nycrophorus investigator. So you can see the line has a positive slope, and it's a significant positive slope. So that shows that in years where there's more rodents, the next year there's going to be more beetles. And if a year is very, very low in rodents, then that means the beetles are not successful at finding a carcass. There weren't very many carcasses available. The number of beetles that were able to reproduce was low. That means next year the beetle population will be small. In a year with a lot of rodents, lots of beetles get to reproduce. They each have good broods. And the next year when they emerge and we capture them, there'll be a lot of beetles. So that's kind of a cool experiment that we've been doing over the years. And that is, again, from doing these year to year, every year we go out and we census these same plots for the rodents and the beetles. And so we're able to only see that pattern because we have many, many years of data, because you only get one data point a year, actually, right? Number of rodents, number of beetles. So it takes a number of years to get that pattern to show up.
And how do you know how many rodents there are in a year?
So we do live trapping, which allows us to do a mark recapture method. So we set a special kind of trap that allows the rodent to go into the trap and be alive and get food and bedding inside of the trap. And then in the morning, we check the traps, we mark them, weigh them, and measure them, find out what species they are, and then we release them right at the site of capture. Our handling of the rodents takes about less than a minute per rodent, and they live to see another day. So we're able to actually, from that mark recaptured data, we're able to estimate the population size of the species. So, because of course we don't want to like snap trap or take the animals, because we want to know what the background number of animals is. So that's why we use live trapping.
Awesome. Does anybody else have any questions about burying beetles? No? I know one thing that we asked of Caitlin, this is probably going to be hard for a burying beetle, so maybe it'll have to be like a mouse joke. Yeah. So Caitlin gave us her favorite squirrel joke. Do you have a favorite species related joke for your system?
Oh yeah. So couple beetles are moving through the airport and they're ready to check in. and they're dragging something with them. And the person at the airport worker is like, do you need to check that? And they're like, no, it's just carry-on baggage. And they're dragging like a dead mouse.
Daniel wanted to know, do they only use a rodent when breeding?
So on this graph, you can see that there's a variety of rodents. So there's a deer mouse, there's a jumping mouse, there's a vole. They use any species of mouse that's available that's within the right size range. So they are kind of picky on terms of size. It can't be too small. Like they usually don't use a shrew, which isn't even a mouse anyway, but it's too small, or a really small mouse. And they also can't bury a really large mouse. So, like a pocket gopher is kind of too big. A golden mountain ground squirrel is too large. Chipmunk's just about the biggest that they could bury, and a large vole. So they are limited on the size, but they don't seem to really care about the species. Some species of burying beetle, but not the ones we work on, are well known to bury snakes and turtles, neither of which we have in abundance in our site in Colorado. And then some are specialists on seabirds, so live on offshore seabird colonies and seem to be able to bury like dead gulls on these big gull colonies and other bird colonies. So there are some burying beetles that are do bury reptiles as well as birds, but we have not found that to be a major source of carcasses at our sites. But at other locations where those organisms are really abundant, and they seem to be able to use them. You can find them feeding on any kind of carcass. So we found them on any kind of roadkill. You'll find them on a deer or a marmot or a skunk. They'll feed on anything, but they won't breed on anything. They will only breed on something that is pretty fresh and that they can bury. So if you're just out there capturing them, you'll find them on pretty much anything that's meat, but they're much more selective in terms of breeding.
And how long do they live, and how many times do they reproduce in their life?
So we don't actually have the answer to that question. We are fairly sure that they can live, we know they can live from the time that they're born, let's say in July, through the winter and through the whole following summer to September. So that's probably the most common life cycle, is to have the summer that you're born and then the summer after this, in other words, the summer you're an adult. So sort of 2 summers or 2 seasons that you could be alive. But whether they can successfully overwinter as adults, some species, that seems to be more common in the species that we've been studying, it seems like overwinter survival is possible, but not common. So we do mark the beetles that we capture in our censuses, and we mark them for the year, and we have caught beetles from year to year that were marked in the previous year. So we know they can overwinter as adults, so they possibly could be active as adults for 2 summers, but we don't have a lot of good records on that. It's very hard to track individual beetles through their life. In captivity, they can live for 2 years, but even that is fairly rare. As an adult, living 6 to 8 months would be a pretty long time. In terms of the second part of the question, the number of broods that they can have, in the laboratory, if you let them rest and feed between broods, they can have 4 to 5 broods. But in the wild, it seems as if they're probably lucky if they get 1 brood, although it seems like they could conceivably have time and the ability to find a second carcass. Whether they get more than that, again, it's unknown because we haven't followed individual parents in the wild. That would be a great experiment to be able to do, but we don't have a really good way to follow an individual from one breeding opportunity to the next.
Do you know about how far that one beetle might travel in its lifetime? And I know they fly, so.
Yeah, so they can fly several miles. We have definitely caught them between our sites that are, several miles apart. The longest distance I know is tens of miles that people have found. But in terms So they don't have, like, unlike other animals that you might be more familiar with, they don't really have like a home range. Like they don't have a place that they live that's like their territory. So their main goal is, when they're an adult, is to find food and to find a mouse to breed on. So they just pretty much fly crosswind, smelling all the time. With those, the antennae are their noses, and so that's what they use to capture particles of smell. And so they're really sensing their environment. They're smelling for the smell of a carcass, and then they land on it. So the further they fly, obviously, the more ground that they can cover, the more likely they are to be able to find a carcass. So they don't, they don't go back to where they were before. They just kind of keep moving. So the chance of us recapturing a beetle is pretty slim.
Okay, any other questions? Awesome. Well, thank you so much. Oh, do they travel together?
Not that we know of. They don't travel together, but they do aggregate. So if one finds a carcass, or even finds your drumstick that you've put in your trap, then they can release a pheromone. And a pheromone is a hormone that is used to change. So a hormone is a molecule that you produce in your body to affect the other cells in your body. A pheromone is a hormone or a chemical that you produce to affect somebody else's body. And in this case, the pheromone that they produce is an attraction pheromone, and it is telling other beetles that they have found a carcass. Males in particular, if they find a carcass and there's not a female present, they release a pheromone that attracts females to the carcass, and then they can form a partnership and breed on it. So if a male finds a carcass, he does release a pheromone. So you often get these aggregations of beetles. So you'll get a whole bunch of beetles finding your drumstick, for example, or finding a carcass. So in that sense, they do communicate with pheromones. And then once they get close, they also seem to communicate with the sounds. But they don't actually travel together. But you do see them bunching up together. They do seem to, be able to find each other.
All right. Well, thank you, Rosemary, for taking time out of your day to talk to us.
Sure, no, this was fun.
Yeah, and we'll put this recording up on our website shortly.
Great, sounds good. Well, thanks everybody, and we'll hope to see some data pouring in of some of the beetles that you've been capturing.
Yeah. Okay.
Anne says thank you too. Everybody have a really great night.
Thanks. Thanks to Erin and Anne for putting on this series. This is going to be really great, I think.
Yeah, it's fun.
Thank you, everybody, for coming. Rosemary, thank you.
Thank you, Anne. They are the